Picture a laboratory somewhere in the early 1970s. A researcher is bent over a comparison of protein structures, hemoglobin specifically, extracted from different species. This is painstaking work done without computers, without genome sequencing, without any of the molecular tools that would arrive in the following decades. And the results coming back are doing something unexpected to the researchers' assumptions. The proteins of chimpanzees and humans are almost identical. Not similar, not comparable. Closer to each other than either is to gorillas, which are supposed to be the chimp's nearest relative by the standard taxonomy of the time. The molecular data is suggesting something that the morphological data, bones, muscles, posture, skull shape, behavior, had completely obscured. That chimps and humans, despite looking so different, despite one of them building cities and the other living in forests, are biologically closer to each other than horses are to zebras. The number that eventually emerged from this work, refined over decades of increasingly sophisticated molecular analysis, is the one you've almost certainly heard: 98.7%. The genetic similarity between humans and chimpanzees measured at the DNA sequence level. Humans and chimps share roughly 98.7% of their DNA. And ever since that number started circulating in popular science, people have been doing something very understandable with it. They've been misreading it in two opposite directions simultaneously. Some people hear 98.7% similar to chimps and feel vaguely insulted, as though the finding diminishes something important about human uniqueness. Others hear it and go too far in the opposite direction, assuming it means chimps are basically people in different packaging, or that ancient humans were essentially chimps who happened to stand up, or that the difference between us and our closest living relatives is so small it barely counts. Both reactions are wrong, and understanding why they're both wrong turns out to be one of the more interesting intellectual exercises in all of biology, because it forces you to actually grapple with what genetic similarity means, what it doesn't mean, what the actual evolutionary relationship between humans and chimps looks like, and why a 1.3% difference in DNA can produce outcomes as dramatically different as a chimpanzee and a person writing a script about chimpanzees. The story is better than the number, so here is the actual story. First, dismantle the number itself, because 98.7% is simultaneously true and misleading in ways that require explanation. When researchers compare human and chimpanzee genomes, they get different similarity figures depending on what exactly they're comparing and how they're counting. The 98.7% figure refers to single nucleotide differences, positions in the genome where a single DNA base differs between the two species. By this metric, at the level of individual DNA letters, humans and chimps are indeed about 98.7% identical. But genomes are not just sequences of single letters. They also vary in larger structural ways: insertions, deletions, duplications, inversions of whole sections. When you account for these larger structural differences, the similarity figure drops. Some analyses put the total genomic similarity, accounting for insertions and deletions, closer to 95%. Others produce different numbers depending on methodology. The honest answer is that how similar are human and chimp genomes doesn't have a single clean answer. It has a range of answers depending on what you measure and how. More importantly, similarity in DNA sequence does not translate linearly into similarity in biological outcome. The relationship between DNA and the organism it produces is not like the relationship between a recipe and a dish, where changing one ingredient produces one proportional change in the result. It's more like a programming language where a single character change in a critical line of code can crash the entire program, while changing 100 characters in non-functional regions produces no visible effect at all. Where in the genome differences occur matters enormously. A difference in a regulatory region that controls how dozens of genes are expressed can have consequences vastly larger than a difference in a coding region of a single gene. The architecture of the genome means that small percentage differences can produce large biological differences, depending entirely on which parts of the genome those differences affect. The human and chimpanzee genomes differ in some very consequential places. And those places are where the 1.3% is doing almost all of its work. Now go back further, because the 98.7% similarity figure is really a statement about the present, about the genomes of modern humans and modern chimpanzees. The question of what ancient humans were in relation to ancient chimps requires understanding the evolutionary history that produced both. The common ancestor of humans and chimpanzees lived somewhere between 6 and 8 million years ago. There's scientific debate about the exact timing, with different molecular clock analyses and different fossil interpretations producing different estimates. But 6 to 8 million years is the current best range. This ancestor was not a chimpanzee. It was also not a human. It was something else. A primate that belonged to neither lineage because neither lineage existed yet. This is where popular representations of human evolution consistently mislead people. The March of Progress image, the famous sequence from stooped ape to upright modern human, implies a linear transformation, as though evolution was a directed project with a destination in mind. It wasn't. When the ancestral population split into the lineage that would eventually produce modern chimps and the lineage that would eventually produce modern humans, both branches were beginning new evolutionary journeys. The chimp lineage evolved. The human lineage evolved. They both changed from the common ancestor in different directions for 6 to 8 million years. Modern chimpanzees are not living fossils of what ancient human ancestors looked like. They are the product of 6 to 8 million years of their own evolution, adapted to their own environments, shaped by their own selective pressures. The idea that chimps represent an earlier stage in human development, a kind of evolutionary rehearsal for what eventually became us, is one of the more persistent and more misleading framings in popular evolutionary discourse. What the fossil record of the human lineage actually shows is a story considerably more complex and surprising than any simple narrative accommodates. The earliest members of the human lineage, genus Australopithecus and related forms, appear in the African fossil record beginning around 4 million years ago, with possible earlier relatives pushing that back further. These were upright walking primates with brains only slightly larger than those of modern chimps, with faces that were distinctly more ape-like than modern human faces, living in the forests and woodland savanna mosaics of eastern and southern Africa. They were bipedal, which is the defining feature that marks entry into the human lineage in the conventional sense. Bipedalism, walking upright on two legs as the primary mode of locomotion, is what separates the hominin lineage from other apes anatomically, and it appears to have evolved long before the dramatic brain expansion that most people associate with becoming human. Lucy, the famous Australopithecus afarensis specimen from Ethiopia dating to about 3.2 million years ago, walked upright but had a brain roughly the size of a grapefruit, comparable to a modern chimpanzee. So for several million years, the human lineage was bipedal apes with small brains. Not chimps, not humans, something distinct from both. They made stone tools beginning around 3.3 million years ago, possibly earlier, which is a behavioral capability that places them outside chimp behavior in an important way. Though modern chimps do use tools in ways that have complicated assumptions about tool use as a uniquely human characteristic. The brain expansion that characterizes the genus Homo, which appeared roughly 2.5 to 3 million years ago with early species like Homo habilis, represents an evolutionary departure that is one of the most dramatic and rapid changes in the history of vertebrate brain evolution. From the small-brained Australopithecus to modern Homo sapiens, hominid brain volume roughly tripled over the course of about 3 million years. 3 million years sounds long in human terms. In evolutionary terms, for a change of that magnitude in a structure as metabolically expensive as the brain, it's remarkably fast. The question of what drove this brain expansion is one of the genuinely contested and fascinating questions in paleoanthropology. The leading hypotheses involve some combination of dietary change, specifically increased meat and cooked food consumption providing more caloric density to fuel a metabolically expensive brain, and social complexity, where larger and more complex social groups created selective pressure for greater cognitive capacity. The two hypotheses are not mutually exclusive. Diet may have made large brains energetically possible, while social pressure made them beneficial enough to be selected for. The brain expansion likely built on itself too. Bigger brains enabled more complex social and technological behavior, which created more selective pressure for bigger brains. Now, here is where the chimpanzee comparison gets most interesting. Because, by the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. Because modern chimps are not simply sitting still as a contrast point. They are sophisticated animals whose behavioral complexity continues to reveal new dimensions that complicate comfortable assumptions about the human-chimp divide. Chimpanzees use tools. This has been known since Jane Goodall's observations in the 1960s, which caused enough of a stir that the scientific community initially pushed back against her findings. The prevailing assumption was that tool use was a defining human characteristic. Chimps strip leaves from twigs to fish termites from mounds. They use stones as hammers and anvils to crack nuts. Different chimp populations across Africa have different tool use traditions that don't appear to be individually reinvented, but are instead passed between individuals through observation and practice, which means chimps have cultural transmission of technology. Different populations use tools differently, maintain those differences across generations, and the differences map to geographic regions rather than to individual learning. That's culture, not human culture, but a meaningful form of cultural transmission that places chimps in a different category from animals that learn only individually. The implications of this for understanding the evolutionary origins of human culture are significant. It suggests the cognitive architecture for cultural learning predates the human lineage and was present in the common ancestor, which means the elaborate human capacity for cultural accumulation may have evolved through elaboration of existing primate capabilities rather than from a completely new departure. Chimpanzees have theory of mind, the ability to attribute mental states to others and to reason about what others know, believe, or intend. The degree and sophistication of chimpanzee theory of mind compared to humans is actively debated, but the basic capability is present and documented. Chimps engage in deception. They conceal food from dominant individuals. They use false cues to mislead competitors. They distinguish between observers who can and cannot see them when making behavioral decisions. These behaviors require modeling the mental states of other individuals. They are not reflexive responses, they are strategic. Chimpanzee social structure is complex, politically sophisticated in ways that primatologist Frans de Waal documented extensively, involving coalition formation, alliance maintenance, status competition, reconciliation behavior, and something that looks behaviorally indistinguishable from empathy in certain contexts. Chimps console individuals who have experienced distress. They share food, particularly after cooperative hunting. They maintain long-term social relationships that influence outcomes across years and decades. None of this makes chimps human. The differences are enormous and they matter. Human language, specifically the capacity for recursive, generative grammar that allows unlimited novel sentence construction, has no parallel in chimpanzee communication despite decades of language research with chimps in captive settings. Chimps taught sign language or symbol systems produce impressive results, but do not spontaneously combine symbols in the recursive, structure-dependent way that characterizes even the simplest human language. The gap between what chimps do communicatively and what humans do communicatively is vast and represents one of the most significant biological differences between the two species. Human cumulative culture, the ability for each generation to build on the technological and conceptual achievements of the previous generation, producing cultural change that accelerates over time, has no chimpanzee equivalent. Chimp tool traditions are stable across generations rather than progressive. They don't improve on the termite fishing technique over time in the way that human technology consistently builds on itself. The cognitive mechanism that enables cumulative culture in humans, sometimes called the ratchet effect, appears to be absent or dramatically limited in chimps, and this single difference has been sufficient to produce outcomes as divergent as a chimp population in a Tanzanian forest and a human civilization capable of writing about chimp populations from a different continent entirely. The relationship between humans and chimps is further complicated by the discovery, through ancient DNA analysis, that the story of early human evolution was considerably messier than a simple linear progression. Analysis of ancient human genomes has revealed that early modern humans interbred with Neanderthals, a closely related human species that diverged from the Homo sapiens lineage roughly 500,000 years ago. And with Denisovans, a group known primarily from DNA extracted from a finger bone found in a Siberian cave. Modern non-African humans carry roughly 1 to 4% Neanderthal DNA. Some populations in Southeast Asia and Oceania carry additional Denisovan DNA. The human lineage in the period before modern humans were fully established was not a clean single species progressing towards the present. It was a complex of related populations with fluid boundaries, occasional interbreeding, and a diversity that the fossil record has only partially captured. This complexity doesn't change the relationship to chimps. Chimps and humans haven't interbred since the lineage split 6 to 8 million years ago, and there's no evidence of gene flow between the two lineages during the period of human evolution. But it does complicate the idea of a simple human type that descended cleanly from a chimp-like ancestor. The actual history is more branching, more reticulated, more full of dead ends and occasional mergings than any linear narrative captures. The genetic differences between humans and chimps that produce the most dramatically different outcomes are worth examining specifically, because they illustrate how small percentage differences can do enormous biological work in the right genomic locations. FOXP2 is a gene involved in the fine motor control needed for speech production, among other functions. The human version of FOXP2 differs from the chimpanzee version at 2 amino acid positions. 2 changes. In the context of a genome with billions of base pairs, 2 changes is essentially nothing. But those 2 changes in FOXP2, along with changes in the regulatory regions controlling its expression, are associated with aspects of the human capacity for spoken language that chimps lack. People with disrupted FOXP2 have severe language and speech impairments. Introducing the human version of FOXP2 into mice produces measurable changes in their vocalizations and fine motor control. 2 amino acid differences in the right gene contributing to outcomes as profound as human language. HAR1, Human Accelerated Region 1, is a section of the human genome that is nearly identical across all mammals but has changed dramatically in the human lineage specifically. HAR1 is expressed in neurons during human brain development, specifically during the period when the cerebral cortex is establishing its characteristic folded, expanded structure. The dramatic difference between the human HAR1 and the equivalent region in other mammals, including chimps, suggests that changes in this regulatory region contributed to the dramatic expansion of the human cerebral cortex during hominin evolution. A regulatory region, not a protein-coding gene, but a stretch of DNA that influences when and where other genes are active, produced changes in brain development that contributed to the organ responsible for human consciousness. Language, technology, art, and the ability to be curious about our own evolutionary origins. This is where the 1.3% lives. Not spread evenly and inconsequentially across the genome, but concentrated in regulatory regions, developmental control sequences, and critical genes where small changes produce cascading consequences through development. The difference between a human brain and a chimpanzee brain is not primarily a difference in which genes exist. Most of the same genes are present in both species. The difference is largely in how those genes are regulated. When they turn on during development, how strongly they're expressed, in which tissues they operate, and how they interact with other genes in developmental networks. So, were ancient humans 99% chimp? The answer requires dismantling the question before answering it. Ancient humans, meaning the early members of the hominin lineage, the Australopithecines and early Homo, were not chimps. They were not 99% chimps. They were early hominins, distinct from chimps from the moment the lineage split 6 to 8 million years ago. Walking differently, probably feeding differently, facing different selective pressures, evolving in their own direction. The genetic similarity between humans and chimps reflects shared ancestry, not shared identity. It means we came from the same place. It doesn't mean we ever were the same thing. The 98.7% figure is real and meaningful. It tells us something important about evolutionary recency, about how recently the split happened, about the degree of shared biological infrastructure that underlies both species. But treating it as evidence that ancient humans were basically chimps confuses the map for the territory. The map says the DNA is similar. The territory, the actual biological, behavioral, and ultimately civilizational outcomes, is where the consequences of the 1.3% become fully visible. The chimp in the forest and the person watching a video about that chimp are separated by 6 to 8 million years of independent evolution, by a brain expansion that has no precedent in mammalian history, by language, by cumulative culture, and by a small but extraordinarily consequential percentage of a genome's worth of regulatory and coding differences that were exactly enough to produce, on one branch of the family tree, an animal that builds cities and writes music and sends probes to the outer edge of the solar system. The chimp, meanwhile, is doing something in a forest in Tanzania that it would be doing whether or not anyone was paying attention, which is a form of biological success that deserves its own respect. The chimp is not a failed human. The chimp is a chimpanzee, an extraordinarily sophisticated primate that has been successfully chimpanzee for 6 to 8 million years and counting, whose cognitive complexity we are still working to fully understand, and whose evolutionary relationship to us is one of the most interesting facts in all of natural history. The number is 98.7%. The story is considerably bigger than the number.